Welcome, and thank you for attending this presentation. My name is Gage Greening, and I'm the CTO of Nanomatronix. Today, I will be discussing AvIDT, which is our novel platform for profiling tissue and cell-based immune responses to injected test materials. Our mission here at Nanomatronix is to use nanotechnology, microelectronics, and biotechnology to provide solutions to the healthcare, energy, and defense industries. Additionally, we are a technology development and transfer consortium that helps transition promising academic research into the applied commercial realm. Nanomatronix is a small business located in Fayetteville, Arkansas, and have been in business since 2005. We are located at the Genesis Technology Incubator at the Arkansas Research Technology Park and are very closely affiliated with research at the University of Arkansas. For this technology in particular, we have partnered with Dr. Gisela Erf at the Poultry Science Department. As a snapshot of a few of our ongoing projects and commercialization efforts, we at Nanomatronix perform screening and qualification of hybrid monolithic circuits, as well as characterization of optoelectronics components for harsh environments, and have ongoing contracts with the Department of Defense and Industry. We're also developing organ-on chips for the Air Force for their Airmen on Chip initiative. We're ruggedizing fiber optic transceivers for harsh environments, developing ultra-long nanofibers and nanotubes, and ramping up our cell and tissue immunological screening platform for the poultry industry. And so this is a quick look at our team here. Dr. Gisela Erf is the original inventor of this technology and serves as a technical advisor to Nanomatronix. And so jumping into things, infectious diseases represent around 1/3 of the issues faced by the poultry industry, and fundamental knowledge regarding events leading to effective immune response and memory development in poultry is lacking. To address this issue, we have developed the Avian Immunology Dermatological test, known as AvIDT. This method for AvIDT was patented in 2012 to study innate and adaptive cell and tissue immune responses. AvIDT is a novel in vivo cutaneous test using the growing feather, a skin derivative, as a platform to analyze tissue and cellular responses to injected test materials. AvIDT involves intradermal injection of test materials into the pulp of a chicken's growing feathers. AvIDT will address the lack of methodology for analyzing the complete time course of the innate and adaptive cellular and tissue immune response from initiation to resolution within a single individual following the injection of test materials. So the primary innovation is the use of the avian growing feather. The living portion or pulp of growing feather is a column of skin around 10 millimeters in height and 2 to 3 millimeters in diameter, and consists of an innerdermis enveloped by an epidermal layer and connective tissue sheath. The vascular supply runs through the dermis of the pulp, allowing for recruitment of soluble factors and leukocytes from circulation. Each growing feather pulp constitutes a distinct unit of tissue and can therefore be considered an in vivo test tube. In other words, there is no cross-reactivity between neighboring feathers. Additionally, AvIDT uses an outbred animal population to capture a wider spectrum of biodiversity uncommon to a majority of animal studies. So importantly, we can use up to 24 growing feathers in a single animal, meaning we have 24 data points per animal. And I'll touch a little bit more on that a little bit later. So what can we inject into growing feathers? And really, it's whatever you are interested in looking at. We can inject both viral and bacterial pathogens, vaccine antigens, adjuvants, nanomaterials, chemicals, and other materials to see how the avian immune system responds to these test materials. And so what are we able to quantify? One of the advantages of AvIDT is that we can acquire a lot of data in a single experiment on a single animal. So we can pretty much look at any cell type, including B cells, T cells, monocytes, heterophils, and macrophages, and can also look at cytokine expression, for example, pro and anti-inflammatory cytokines, antibodies, and reactive oxygen species. Each of these biometrics can be quantified per growing feather. So what is the actual process? If we are interested in developing a new vaccine, for example, the key stages of AvIDT include the plucking of the 12 growing feathers in a row along each breast tract, which is painless for the animal, vaccination of the animal against this test material, plucking the growing feathers and waiting 18 days for the growing feathers to reach the optimal size to inject about 10 microliters of test material. And so then we're able to inject the test material into the multiple growing feathers, as shown here, And then we're able to collect these growing feathers at different time points on a wide scale of minutes to weeks. And so then we can take these pulps, extract the pulps from these extracted growing feathers, and undergo ex vivo examination of the growing feather pulps to quantify the biometrics listed on the previous slide. And so once the pulps of the growing feathers have been extracted, we can run several ex vivo tests, including histology and immunohistochemistry, flow cytometry for phenotypic cell population analysis, as well as qPCR for gene expression analysis of key chemokines and cytokines. We can also run various blood tests for blood antibodies. So what really makes AvIDT unique? First, we can inject up to 24 growing feathers in a single bird, meaning that experiments are highly customizable. Each individual growing feather can be injected with different test materials, different concentrations of test materials, and can be collected at different time points ranging from minutes to weeks. Thus, we can study the complete time course of the innate and adaptive cellular and immune response from initiation to resolution within a single individual following injection of test materials. This is then repeated in multiple animals according to the power analysis of your study. So next we'll get into some sample data. So here each data point that you see is the average of multiple growing feathers. We can see different leukocyte cell types on the y-axis and time on the x-axis in response to being injected with a PBS control or a graphene-based nanomaterial. So this study in particular was looking at nanotoxicity. So in the top left image, we can see a spike in leukocytes at day 1 with a gradual drop-off. Next, in a different study, we see T and B cells on the y-axis with time on the x-axis and in response to chickens receiving PBS compared to those receiving injections of mycobacteria. One cohort of chickens was first immunized against mycobacteria, whereas the other was not. Next, in the same study as the previous slide, we can see different cytokines, different interleukins, interferons on the y-axis as a function of time in both vaccinated and non-vaccinated chickens after injection with mycobacteria as the test material. And as stated earlier, we can also perform histology and immunohistochemistry. So here we have stained for T cells and B cells alongside a graph showing how these lymphocyte populations change over time after injection of test material in vaccinated and non-vaccinated animals. So AvIDT has several broad applications, including vaccine development and optimization, studying the effects of diet immunomodulators, immunotoxicity, and nanotoxicity. And these studies can also be complementary with data obtained in preclinical ADME studies. So recently, for example, diet immunomodulators have been a hot topic. So as an example, AvIDT could be used to see how diet affects the immune response after injection to a pathogen. Regarding vaccine development, AvIDT could be used to compare the immune response to a target pathogen after being immunized with your new vaccine compared to your old vaccine or potentially a competitor's vaccine. So with that, I'd like to acknowledge our entire team. Dr. Gazella Irf is the original inventor of AvIDT, and our biomedical engineer, Ms. Rodriguez, is the technical expert who gets her hands dirty with these chickens, collects and analyzes data, and really does the majority of the actual hard work here. So right after this presentation, Mr. Parker Cole, our Chief Scientific Officer, will be answering all of your questions. And with that, thank you all for listening in.